Okay, let's unpack this. We like to think of modern medicine and, especially cancer treatment as this super precise science.
Laser focused, right?
Exactly. You get a diagnosis, there's a specific pill.
Yeah.
Problem solved. But if you actually talk to an oncologist, you realize there's still a frustrating amount of, guessing.
It's the classic black box problem. I mean, we have these incredibly powerful drugs now, especially immunotherapies, that they can melt tumors away for some patients. It's miraculous.
But for others?
For others, absolutely nothing happens. And for the longest time, we haven't really understood why one person gets a miracle and, the person sitting right next to them in the clinic gets nothing.
That is exactly the mystery we are diving into today. We're looking at a landmark study from Nature in 2023 that really tries to crack open that black box for triple-negative breast cancer.
Which is a notoriously difficult beast to tame.
It is. But what really caught my eye about this paper isn't just the cancer type, it's the method. They stopped looking at the tumor as like a list of ingredients and started looking at it as architecture.
Right. The paper's title is Spatial Predictors of Immunotherapy Response in Triple-Negative Breast Cancer. And that one word, spatial, is doing so much work there.
It's everything.
They're treating the tumor like a neighborhood. It's not just who is there, but who is standing next to whom and who is talking to their neighbor.
It's a detective story, but at a cellular level. We're zooming in to find clues that tell us before a patient spends months on a grueling treatment, if it's even going to work. So before we map this neighborhood, we have to understand the territory. Why triple-negative breast cancer? Why is it the target here?
Well, to get why it's so aggressive, you have to know what it's missing. Most breast cancers have receptors on the outside of the cell, like little docking stations. Docks for hormones like estrogen or for a protein called HER2. And if a cancer has those, we have drugs that can grab onto them.
Those are the handles we can use.
Exactly. But triple negative means the cancer tests negative for all three of those. It doesn't have the handles. So for a long time, all we had was chemotherapy, which is, it's a blunt instrument.
Until immunotherapy came along.
Right. Immune checkpoint blockade, or ICB. This is the big revolution.
The Nobel Prize-winning stuff.
That's the one. The drug in this trial, that trial, is called atezolizumab. And the way it works is, your immune system has soldiers called T cells. Their job is to kill cancer.
Okay.
But cancer cells are clever. They can put up a protein on their surface called PD-L1. Think of it like a secret handshake. When the cancer cell shakes hands with the T cell, it sends a signal that basically tells the T cell to go to sleep.
It's a stand-down order. It deactivates the police force.
That's the idea. So atezolizumab, the drug, is an antibody that physically gets in the way. It jams that handshake. It stops the sleep signal from getting through so the T cells stay awake and do their job.
But here's the catch, and the whole reason we're doing this deep dive. It only works for some people. And this trial wanted to figure out who, using a technology called imaging mass cytometry, IMC. How is that different from, just looking at a biopsy slide?
It's a complete game changer. So traditional pathology is like looking at a blurry satellite photo. You see the forest, maybe some clearings, but not the individual trees.
Okay.
Then you have something like genetic sequencing. That's like taking the whole forest, throwing it in a giant blender, and making a smoothie. You know exactly what's in there, how much pine, how much oak, but you've lost all the structure. You don't know who is next to whom.
And in biology, that structure is everything.
It's everything. So IMC is it's like Google Earth for the tumor. They take the biopsy, but instead of using dyes, they use antibodies tagged with heavy metals. Then they hit the tissue with a laser, pixel by pixel, and they can map 43 different proteins at the same time.
43. So you can see not just where every cell is, but what it's doing, who it's talking to.
At a subcellular level, it's an incredible map. And the most crucial part of the study design was the timing. They took biopsies at 3 points. Baseline, so before any treatment.
The starting point.
Then on treatment, so right after the first cycle. And then post-treatment at the time of surgery.
So you're not just getting a snapshot, you're getting the whole movie.
You get the whole movie.
Okay, so let's hit play. They start at the very beginning, that baseline biopsy. The big question. Can we look at this pregame map and predict who's going to be cured?
Which is the holy grail. The clinical term is pathological complete response, or PCR. It means the surgeons go in and find zero cancer left.
Wow. And what they found was that it's not enough to just count the immune cells. You can't just say a tumor is hot and call it a day.
No, it's not about quantity. It's about quality. It's about who the cells are. And they found two huge predictors at baseline. First, it's about the immune system's bench strength.
Okay.
The patients who got a cure started out with a very specific T cell, CD8 plus TCF1 plus T cell.
Whoa, okay, that's an alphabet soup. Let's break that down. CD8s are the killer T cells, right?
They're the cytotoxic ones, yes. But that TCF1 marker is the key. TCF1 signals that the cell is stem-like.
Stem-like, so they're not actually fighting yet?
Not yet. They're not the ones on the front lines with their guns drawn. They are the progenitors, the source.
So they're like the reserves, the reinforcements.
That is a perfect analogy. Think of them as the sleeping army waiting in the barracks. The study found that having a big, proliferating army of these TCF1 plus cells at the start was the single strongest predictor of success.
That seems a little backward, though. I mean, wouldn't you want the active killers there from day one?
You'd think so, but immunotherapy is a marathon, not a sprint. If you only start with the active cells, they burn out, they get exhausted. You need this deep pool of TCF1 plus cells that can keep dividing and spawning new waves of killer cells once the drug takes the brakes off.
So if you don't have the reserves in the barracks, you can't win the long war. That makes sense. Okay, that's condition number one. What's the second?
The second condition is about the cancer cells themselves. Patients did way, way better if their cancer cells were expressing something called MHC-II.
MHC2. Wait, that's normally on immune cells, isn't it? It's the flag they use to show off threats to each other.
It is. Major histocompatibility complex class 2. It's usually on what we call professional antigen-presenting cells. But sometimes, for reasons we don't fully understand, cancer cells start expressing it.
Why on earth would a cancer cell do that? Isn't that like painting a giant target on your own back?
That's effectively what it's doing. It's making itself highly visible. When a cancer cell puts up MHC-II, it is screaming to the immune system, "Hey, I'm foreign, come get me."
Okay, let me pause you there. I want to do a quick recap before the treatment even starts. So we've got the Tech IMC, which is giving us this Google Earth view of the tumor. And for a patient to have a good shot at a cure,
you need two things at the starting line. One, you need that sleeping army, the TCF1 plus T-cell reserves ready to provide reinforcements.
the reserves.
And two, you need the enemy to be visible. You need the cancer cells to be flying that MHC-II flag.
Reserves and visibility. Got it. And you're saying the study showed these things were happening in the same place.
That's the spatial part. They found that TCF1 plus T cells were often physically touching the MHC-II plus cancer cells. They were already in the right neighborhood, ready to go.
So the stage is set, the whistle blows, treatment starts, patient gets one cycle of chemo and immunotherapy. 3 weeks later, they go in for that second biopsy, the on-treatment look.
And this is where the static map becomes a dynamic battlefield. For the patients who are responding, the landscape just transforms.
What happens to that sleeping army?
They wake up. The number of TCF1 plus cells actually drops. And in their place, you see this massive, massive surge in CD8 plus GZMB plus T cells.
GZMB, granzyme B.
Granzyme B. It's a cytotoxic molecule. I mean, it is literally the poison bullet that T cells fire into cancer cells to make them explode.
Oh.
So the sleeping army has become the fighting army. The responder maps just light up with granzyme B. It's an active war zone.
Okay, that's the best case scenario. Reserves deploy, bullets fly, cancer dies. But what about the patients who don't respond? What does a resistant tumor look like?
This is where the paper reveals this really fascinating villain. In the tumors that were resisting treatment, they found a new player on the field. Cancer cells that were expressing CD15.
CD15. Hang on. Checking my notes. Isn't that a neutrophil marker? That's a type of white blood cell.
It is. It has absolutely no business being on a breast cancer cell. It's a carbohydrate antigen you find on neutrophils.
But what is it doing there? Is the cancer wearing a disguise?
It really looks that way. It's a form of mimicry. And spatially, it was very distinct. These CD15 plus cancer cells appeared in what they called a mosaic pattern.
A mosaic?
Yeah, you'd see a patch of normal cancer cells, and then mixed in like tiles are these weird CD15 plus cancer cells. And here's the kicker. Sometimes these clusters were surrounded by actual neutrophils.
No way.
It suggests the cancer is undergoing some kind of state change, maybe to hide, maybe to recruit those neutrophils as bodyguards.
That is incredible. It's a wolf in sheep's clothing. The cancer puts on a CD15 coat, hangs out with the neutrophils, and the killer T cells, the granzyme B squad, They just can't seem to touch them.
And the data was clear. The presence of these CD15 plus cancer cells, even after just one dose of treatment, was a huge red flag. If you see CD15 go up and you don't see that granzyme B surge, the treatment is failing.
There was one other character in those resistant tumors, plasma cells.
Yeah, that was another interesting finding. Plasma cells are mature B cells. They make antibodies, usually good guys.
Yeah.
But in the resistant tumors, they found these plasma cells physically touching the cancer cells.
So in this specific neighborhood, they might actually be collaborators, helping the cancer hide.
It's possible. It really drives home the point that in biology, context is everything. A cell that's good in one situation might be bad in another.
Okay, let's freeze frame again because that CD15 finding is a really big deal. So we're now looking at the biopsy taken during treatment. The good sign is seeing your TCF1 plus reserves deploy into a swarm of granzyme B killers. The map should look like a war zone.
Yes. The bad sign is the appearance of these CD15 plus cancer cells. It's a sign that the tumor is changing its identity to escape.
So you have the baseline data and you have the on-treatment data. The researchers then combine them. Why was that so important?
This is really the so what of the whole study. They built a predictive model. Using just the baseline data, the reserves and visibility, they could predict who would be cured with about 77% accuracy.
Which is pretty good, but not perfect.
Not perfect. And using just the on-treatment data, that granzyme B versus CD15 battle also got them about 77% accuracy. But when they combined them,
Let me guess, it went up.
it jumped to 82% accuracy.
So you need both. You need to know the potential at the start, and you need to see the actual reaction to the first punch.
Exactly. Which leads to the most practical, powerful idea in this paper, adaptive therapy.
Because right now, we give the treatment, and we just keep giving it for months and hope it works. We wait for a scan way down the line.
And that's lost time. This study suggests a whole new way of doing things. Imagine a patient comes in. We take that first biopsy. We see they've got the TCF1 plus reserves. Great. Start treatment.
But then, 3 weeks later, after just one dose, you go back in for another tiny biopsy.
And if that second look shows a huge surge of granzyme B, fantastic. Stay the course. But if it shows those CD15 plus cancer cells popping up and no killing, we know with 82% certainty this isn't working.
So you stop, you switch to a different chemo, you try a different drug, you don't waste another 5 months on a failing therapy. That is true precision medicine.
It changes the entire paradigm from wait and see to check and adjust.
I want to go back to that CD15 disguise for a second.
Yeah.
The paper calls it a state transition. Does that mean the cancer is actually changing its DNA?
Probably not its DNA, but its expression program, its phenotype. It's called plasticity. The intense pressure of the drug attack forces the cancer to either evolve or die. And the ones that figure out how to switch on the CD15 program are the ones that survive.
And since they were hanging out with neutrophils, is it possible the neutrophils are teaching them how to do it?
That's a leading hypothesis. We know cells talk to each other. If a cancer cell is getting hammered by T cells and it's right next to a neutrophil, it might just co-opt the neutrophil's survival signals. It's basically saying, hey, let me look like you so the cops leave me alone.
It's just so devious. But if we know CD15 is the shield, maybe we can develop a drug that targets the shield.
Or a drug that stops the cancer from putting on the shield in the first place. This is why this kind of spatial map is so incredibly valuable. It gives us new targets we didn't even know existed.
So the full narrative arc here. We started with a black box, and IMC really turned on the lights.
It did. In the preparation phase, we learned a cure requires a visible tumor, the MHCII+, and a reserve army of T cells, the TCF1+.
Then in the engagement phase, after that first dose, you have to see the reserves deploy. You need to see the granzyme B.
And finally, we learned about resistance. Failure is often marked by the tumor putting on that CD15 disguise to hide.
And the key takeaway is that by combining those two time points, we get a highly accurate prediction. It opens the door for real-time changes in treatment.
It validates the idea that a biopsy isn't just a label you get at the start. It's a monitoring tool, a status report from the battlefield.
Before we wrap up, I want to leave our listeners with a thought that it wasn't explicitly in the paper, but it's nagging at me. If CD15 is a resistance marker, and it's normally on neutrophils, does having a high neutrophil count in your blood just generally make you less likely to respond to this stuff?
That is a fantastic question. And we do know from other studies that a high neutrophil to lymphocyte ratio in the blood is often a bad sign in cancer. This paper could be providing a direct, spatial mechanism for why that is. The neutrophils might be providing the camouflage.
Wild. And a final question for you, the listener. Put yourself in the patient's shoes, that second biopsy. It's invasive, it's not fun, but would you agree to it 3 weeks in if it meant knowing with 82% certainty if the drugs were working, instead of waiting 6 agonizing months to find out?
It's a trade-off. A little discomfort for
Yep.
for invaluable information. I know what I would choose.
I think I'm with you. Knowledge is power, especially when you're fighting a war at the cellular level.
Absolutely.
Well, that wraps up our deep dive into the architecture of a cure. Hopefully you'll never look at a fruit smoothie or a Google Earth map in quite the same way.
And just remember, biology isn't a list of ingredients. It's all about who is talking to whom.
Thanks for listening. We'll see you on the next deep dive.